Hey guys, it's Jordan here. This is presentation 3. We are going to be discussing how oxygen binds to the heme. First off, let's do a little background here on how exactly we are doing this binding and where it is located. So we all know we have our lungs, and inside our lungs we have our bronchioles, which keep breaking down, and then eventually we get to the alveoli. Those are those gray-black structures that do the gas exchange. This is also where the oxygen binds to the hemoglobin, is in the lungs. So within these little alveoli, we have little capillaries, and inside these capillaries are our red blood cells. The red blood cells are where you'll find the hemoglobin. So here you can see a little bit clearer. The red blood cell carries oxygen from the lungs into the rest cells, and then from there the oxygen is released into the tissues. That's more of a general overlook there. But going to a little deeper, further explanation. Here is the picture of our red blood cells. Each cell contains several 100 million hemoglobin molecules. So this here is the hemoglobin molecule, and these are what are carrying oxygen. The oxygen parts are binding to the hemes on the hemoglobin molecule. That took me a little bit to, I thought a heme was a hemoglobin molecule, but anyways, the oxygen is binding to the heme specifically inside the one hemoglobin molecule, and there's so many of these. A little further in. So as you can see, the Heme group is non-planar when the oxygen is not bound to it. The iron atom is out of the plane, and this configuration of deoxygenated hemoglobin is called a doomed shape. You kind of see that there. The regions of the electron density push the other apart, and then the iron atom in the center is drawn out of the plane. On the right side here, we have the oxygenated, and we can see here that when the oxygen is binded to the heme, it will become planar, and the coronary structure of the hemoglobin makes it obvious that changes occur after the oxygen binds to the heme. All right, clear as mud. Okay, and then a little further in depth here, talking about the resting or the relaxed and the taut phases. So in the deoxygenated state, dimers are linked by extensive surface area, and then a lot of the heme groups, they're separated corresponding to the T state. So the T is when they're calling that because it's not, or it is tense, kind of packed together. The oxygenated state is the iron is moving and on the oxygenation, iron moves and brings iron-associated histidine towards the porphyrin. Then you can see the histidine containing the alpha helix that it's altering, and this is what they're calling the rest or relaxed state. Here we can see that 1O2 binds to a hemoglobin substitute or subunit, and then the O2 binding disrupts the inner dimer bonds. See this right here? And then this increases the oxygen affinity or the likeliness of the remaining substitutes. So they're going to want to come together. Carbon dioxide can bind to the hemoglobin subunit because it is a change of the protein that decreases, like I said earlier, it's decreasing the oxygen affinity for the molecule. This is also known as an allosteric inhibition. Also, the carbon dioxide will react with the water, the H2O, which will lead to the creation of bicarb in the hydrogen ion. Then those hydrogen ion protons will reduce the pH of environment. So then that's going to lower the hemoglobin's attraction for the oxygen. All right, if you guys have any questions, feel free to ask. Otherwise, thank you for watching.